I'm Carl Whitcomb, and today I want to share with you an aspect of communication. Early on in my career, in trying to teach students about plants, research, understanding plant responses, I coined the term, "Let's ask the plant." Now, I could have said, let's go set up an experiment. But let's ask the plant kind of personalize it a bit more. And so I've used that extensively, and I still use it today in talks and presentations here and there. The point being that if you ask the plant whether they like treatment A, B, or C, and you set up a simple experiment properly, pretty good chance that you will find an answer to your question. The plants will tell you whether they like treatment A better than treatment B or better than treatment C. And it may come as a surprise to you. One of those early experiments came about from questions in a nursery management class I was teaching at the University of Florida. I had to say I don't know frequently because there was so little known about growing plants in containers out of doors. What had been written mostly was from work in California. Well, if you're in a desert, the only time the plants get watered is when you turn the sprinklers on. Well, if you're in Florida, it may rain, and some more tomorrow, and some more the next day. So drainage and aeration, and aeration is critical to root functions. Big difference. The other thing that was prevalent back in those days were micronutrient deficiencies. I mean, everywhere on almost every species of plant. So after saying I don't know about some of the questions regarding micronutrient fertilizers in class, one of the following laboratories, we set up a simple experiment. And it involved 3 treatments. We simply did a 0 level of micronutrients added to the mix in the containers. We did the label rate, which was a ½ pound per cubic yard of an old product called Perk, and we doubled that to 1 pound per cubic yard. Well, by the end of the semester, clearly the 1 pound per cubic yard was way better than the ½ pound, and of course way better than 0. The plants had spoken. They said, we like more micronutrients. It also begs the question, well, if 1 pound is better than a ½ pound, what about 2 pounds or 4 pounds? So quite often the first experiment begs the second experiment, begs a third experiment if you keep exploring. In Plant Production in Containers, this little book that I wrote a number of years ago and thoroughly revised a few years ago, I describe in the back of that how to set up an experiment. It doesn't have to be complicated. It needs to be replicated, the treatments, randomized, and you control everything except the treatment. So keep everything else uniform. Well, replications, all right, that means 1 replication, 2 replications, 3. I prefer to use 10. Randomization, if you only got 3 treatments, randomization can be pretty simple, and even for larger numbers. For example, if you have treatment 1, treatment 2, treatment 3, then the second replication you go treatment 1, treatment 2, treatment 3, and the third replication you go treatment 1, treatment 2, treatment 3, and repeat that. That's pretty random and scientifically acceptable, statistically acceptable. Then watch the plants and evaluate them by height, color, whatever criteria you're interested in. The key, too, is starting out with uniform plants to begin with. If you have variation among the plants that are in your treatments, you probably stubbed your toe. A lot of this information is in a little book called Plant Production in Containers. I've written 5 books, or Plant Production of Landscape Plants in the Field, or Establishment and Maintenance of Landscape Plants, the Big Plant Identification book. And if you want to know more about the character on this video, it's in this autobiography. So there's lots to think about, but you can communicate with plants. I describe plants as being happy when they're growing well, and that there's a luster on the leaf and they're happy. I sometimes describe plants as being under stress, and you can tell when they're under stress. That means you need to do another study or do some more exploration in terms of finding an answer as to what the restricting factor is. So, the other thing you could do if you're interested in this topic is to go to 2 websites. Rootmaker.com is one. I have lots of papers on that website. And if you just type in my name, Carl Whitcomb, in a Google search, it'll take you to the LaceBark website, and there's lists and lists and lists of papers that I have written, research results presented there. So maybe among these factors, there's some bits and pieces that will assist you.